It now gives me great pleasure to introduce Dr. Schuldiner, a senior scientist in the Institute's Department of Molecular Cell Biology. Also, she is quite the skilled baker, as she applies the same sense of invention and curiosity to her culinary skills as she does to her lab work. Please welcome Dr. Maya Schuldiner.
Good morning. So wonderful to see you all today. I was looking around as you all came in, and I'm already starting to know some of the faces from yesterday. And I would like to thank you for taking your morning and spending it with us. I know that you do it out of love for the Weizmann Institute. And I also think that in a way you share with Ron Jacob and myself a kind of love for the magic of science, because the reason that I do science, and I think the reason that you've all come to hear about our science, is because it is so magical, because we don't stop often enough to remember that everything around us is really exceptional. And for me, the thing that demonstrates this most are my kids. So some of you heard about them yesterday. This is Daniel, he's 7, and Noam is 4. And being a good Jewish mother, I think they're amazing. But they're amazing not because they're different than any other kid. They're amazing because they're identical to every other kid in nearly every aspect of their life. Just like every other kid, they can run, they can smile, they can talk, they can think, they can eat, they can move their body. In this case, they're rafting. Maybe not every kid does that. But, and you have to sometimes stop and look at this and realize that it's incredible. It's incredible that we have a body that is capable of doing all these things in parallel, and not just performing these different functions, but performing them perfectly well over tens and tens of years without, having to replace spare parts or only maybe sometimes stopping to break down. And the reason our body can do that is because it's a commune. And some people don't like to think about it, but actually, our body is made up of many different organs. So we have our heart, and we have our brain, and we have our lungs. And each of those organs is made out of cells, individual cells. We actually have 50 trillion cells in our body. That's just an enormous number. That's 50 million cells. And those cells have to work together all the time. So on the one hand, a cell in your lung has to do its special role in the commune. Only it knows how to do that. It takes up oxygen and moves it into the bloodstream. The cells in your liver have to do their little role in the commune by detoxifying substrates in your food, and the cells in your brain have to do their little role in the commune of transferring information. But as they do that, they're also individuals in this commune, so they have to survive as an individual in this commune. And this community means that they have to communicate, because you cannot hold 50 trillion things, people, cells, whatever it is you want to build a community out of, without having them communicated every single moment. And if they stop communicating for 1 second, everything crashes, because they have to know what they have to make, when they have to make it, when they have to stop making it, how much do they need to grow, when do they have to stop growing. And that all has to work very, very effectively inside our body at every given moment. So let me tell you a bit about how that works. And that works because every cell and every organ in our body is like a little factory. It's like a factory because it produces something that's important for the community of cells, for our body, but it also maintains itself in some way. And let me tell you a bit kind of about my metaphors for how the cells work. So here in the center is the most important part of the cell. That's where the president of the cell sits, the DNA, that has all the information, the ideas on how to run the cell. It has little blueprints on how to build the factory and why are we doing this and what's its projection into the future of how everything is going to be done. But of course, the DNA in the nucleus is extremely important, but could do nothing without the workers in the production lines. So we have ribosomes, these little black dots inside the cells. These ribosomes are the producers of the cell. They make proteins, and the proteins are necessary to allow the cells to do all of their different functions. And they're making an enormous number of proteins all day. And of course, you need power to run a factory. So we have mitochondria. These are these organs here in red, and the mitochondria produce the power necessary to run this factory. They're like the little power plants of the factory. And now comes the part of the cell that I am most interested in, and that's the shipping and packing department. So it's great to be able to produce stuff in your factory, but it's really not very helpful if you can't get it out and have the other parts of the body enjoy it. And so you need a very elaborate place where you can quality control every single protein that comes out of your cell, and you can package it correctly, and you can make sure that it's going to be functional once it's outside, and then you send it to the external part of the cell. And to do that, it has to pass the membrane, which surrounds the cells and keep it as a unique environment and different from the other cells around it. And you may ask yourself, why would somebody be interested in the shipping and packing department? Because, I mean, a lot of scientists try to study, you know, the VP's office or how the fundraising of the cell happens, and, you know, things that seem to be really important for how the cells work. But I'm interested in the shipping and packing department because without it, cells could not communicate with every other cell type. And because the shipping and packing department of the cell, that is called the endoplasmic reticulum, or ER for short, has to deal with an enormous responsibility. So let me try and demonstrate to you how big the responsibility of the ER is. For example, you just had a little breakfast outside, and when you did that, immediately your body had to regulate your blood sugar levels, right? And in order to regulate those blood sugar levels, it had to produce insulin from its pancreas, from its beta cells. And insulin is a protein that is made inside our beta cells and has to be secreted outside of the cell through this shipping and packaging department. And every single beta cell in your pancreas makes 10 to the 7th, that's about 10 million molecules of insulin every single day. Think of the numbers. It's really amazing. More than that, as you're sitting here listening to our talks, your brain is constantly processing information of what I'm speaking, of the lights, the sounds around you. And in order to do that, an enormous number of electrical signals are going through your nervous systems. And the only reason those electrical signals can move through your brain is because of proteins on the cell surface that allow those signals to pass through. And those proteins are made in the ER, in the endoplasmic reticulum. There are 10 to the 17th of those. That's such an enormous number that the endoplasmic reticulum, the ER, of these nerve cells has to function non-stop in an optimal manner to be able to produce them. Let me give you another example that I really love, and that's an example of our immune cells that are important for guarding us against infection. This is a picture, an electron microscope picture. It's a microscope that can see with very high resolution into the cells. And this is a picture of a B cell, a white blood cell, that helps protect our body against invasion of bacteria. And when you take a picture of that cell, it actually doesn't look very interesting. You can see the nucleus here, and you can see other little parts of the cell. And this cell is resting. It's not creating antibody at the moment. But once it meets a pathogen, once it meets a bacteria, it gets activated, and now it has to produce antibody against that bacteria. And within several hours, it turns completely from a cell that looks like this to a cell that now looks like this. And the one thing that you can probably appreciate is that it doubles in size. So it becomes much bigger. It turns on all its production lights, all its production lines, all the power. And it also gets filled up with this lattice inside the cell this is a reticular network. This is the endoplasmic reticulum that I am interested in. And it is this endoplasmic reticulum that now fills the entire cell that allows these to produce their own weight in antibodies every single day. Now think about it. If we could make a factory that would produce its own weight in product every single day, somebody would get very, very rich. Okay? This is really kind of amazing that a cell can do this. This is really spectacular. So when this goes wrong, you can of course appreciate that it's kind of a catastrophe, because the moment the cells cannot communicate, the moment that they can't sense their environment, the moment that they can't respond to it, then our body collapses, then our body cannot function. And it depends where the ER goes wrong, and you can get an enormous variety of different diseases. For example, if the ER does not function in your pancreatic cells, you can get diabetes because you're not secreting insulin properly. If the ER doesn't function in your white blood cells, as I was showing you, you can get immunodeficiencies because you're not producing antibodies properly. And if it's not functioning in the pacemakers of your heart, then you can't transmit electrical signals, and you can get a heart attack. So really, every single cell type in our body has to have a perfectly functioning ER, or else we will get a large variety of diseases. So I would like to understand every single aspect of how this part of the cell, how this organelle works, how it's divided into different areas, into quality control areas, into trafficking areas, that means the shipping part of this organelle, and how it talks with the cell and adjusts itself as the changing needs of the cells develop. And in order to do that, I need to study cells. So I'm a cell biologist, because all cells have an endoplasmic reticulum, all except for bacteria and viruses. So one option would be to study human cells. And Ron showed you, and Jacob showed you, that we can study human cells in a petri dish, in a culture dish. The problem with that is that human cells are very expensive to grow. They're kind of a pain, that they want their own little special conditions that are similar to how they are in the body. And it's actually pretty difficult working with them for extended periods of time. And so what we do is we use the little trick of evolution. And this means that we look at the simplest cell that still has an endoplasmic reticulum, and that is the baker's yeast. The baker's yeast, you probably all know. You heard I'm a baker, but that's not why I'm using it. It's the stuff you put into your bread to make it rise, or into your beer to make the alcohol or wine. But it's also, in an amazing way, a single-celled animal that is nearly identical in every single aspect to every one of the cells in our body. So we can study it in the lab, and we can learn from it on all the different aspects of creating a cell. And for my purposes, for creating an endoplasmic reticulum. And we call this baker's yeast also the budding yeast, and that is because it divides by budding. And you can see here a mother cell, a mother yeast cell, and its little baby daughter cell budding off of it. And it actually does this budding thing very efficiently. It does it every 90 minutes, which is extremely fast. And so this is why we love working with it in the lab. And this is a joke that really exemplifies to me why we do this. It says, "I wish you'd learn to put the lid on your petri dish, Harry. We came here with 4 kids, and now it looks like we've got 20 million. So that's what we have in the lab. We put one cell in our petri dish, and within a day, we can have millions and millions of them, and we can do whatever it is we want in order to start studying them. And this brings me to the tools that we're using. So I told you that I'm a cell biologist. I'm interested in how the cell works. But I'm also a geneticist. And I'm a geneticist because I use the DNA of the cells, the genes, the information inside the cell, in order to understand how the different parts of the cells are built. And the amazing thing about yeast is that we, as geneticists, we can do whatever we want with its genes. It's very easy to change the yeast's genes, much easier than any other animal cell. And so within a couple of days, we can put in any gene that we want, we can take any gene out, we can make chimeric genes, we can change the place or the time that they're expressed. Now you know what expressed is. and we can attack every single aspect of how this organelle is created. And this has been done for many, many generations, and people have asked a lot of extremely interesting questions with yeast and trying to understand the different functions of the cell. But there were types of questions that we could never answer because we didn't have the right tools yet. And I want to tell you about one of those questions that we're trying to attack now in the lab, and that is, how does this endoplasmic reticulum, how does this ER, get transferred on from mother to daughter cells as the yeast is budding? I told you that this is an essential organelle. Without it, no cell can survive. And so every time a yeast cell buds, or every time one of our cells in our body divides, it has to pass on, just like it passes on its genetic information, it has to pass on its endoplasmic reticulum. And nobody knows how that is done. And so we can now take these yeast that you see here, we can label their endoplasmic reticulum with special fluorescent proteins, and that's, that's the picture that you see here. So this is the endoplasmic reticulum here. It looks like two little concentric rings, and this is the little buds coming out of the mother cells, and you can see the ER is being distributed into them. And we want to know how any alteration in the genes changes the ability of these cells to pass on the endoplasmic reticulum. But this means we have to look at thousands and thousands and thousands of microscopic images, and that is very difficult for the human eye to do. And maybe if I had a limitless number of students, I could have had them sit on this project, but it's actually nearly impossible to do this, and this is why nobody has ever done it in the past. But one of the amazing things about the last couple of years, and you've heard a bit from Ron about this, and one of the wonderful things about the Weizmann Institute is that we have the capacities to work with the state-of-the-art tools in our field, meaning robotic tools that allow us to do these experiments in a way that's completely automated, that's completely robotic, in a way that we call high throughput, because we can do the same experiment that a single person would do in the course of a month. We can now use robotics to do thousands of these experiments over the course of a day. And this means that by using these types of tools that I now have in my lab at the Weizmann, a microscope that is completely automated and can take pictures of thousands of different yeast cells a day, each one with different genes being disturbed or added into them under many, many different conditions, with different drugs, if we want to try and see how drugs affect this process, for example. And using these types of robotics that prepare our sample and do it very accurately and efficiently, we can start tackling questions like this that could never before be tackled. And I hope that this will allow us to understand how this very basic machinery that is essential for the function of every single cell type in our body actually takes place during the normal life of a cell and during disease. But it's not enough to have an interesting model organism. It's not enough to have an interesting biological question or great robotics. And actually, the one thing that I've learned during the last years that I've been training in science is that the big difference as to what type of science you could do and what kind of breakthroughs you can perform is only dependent on the type of people that you can work with. Because in the end, it's really about the group of scientists coming together, thinking in a collaborative fashion, coming up with exciting new ideas and exciting new ways to tackle them that allow us to really make a change in our understanding of life. And I've been extremely lucky at the Weizmann Institute to be part of an amazing group of young scientists. I now have 8 undergraduate and graduate students working in my lab. They're just a tremendous pleasure to work with every day, and I see them growing as scientists, learning how to ask the right questions, learning how to use the right tools, learning how to make the next generations breakthrough in science, and it is thanks to these people that I think in the next years we will really be able to change the way that we see cells and how they function. And so this is my group, and everything that I do depends on them and their wonderful abilities. So I really thank you for coming today.